Okay, we now next need to consider protein structure and folding. The first structure and folding we are going to talk about is what's considered the primary structure of a protein. The primary structure of a protein is basically the linear amino acid sequence of the protein. So this is the combined amino acids in their linear form, linked N to C, to form the full length of the protein. That is the primary structure. Now the primary structure has some very interesting properties, chemical properties, stereochemical properties, that we need to consider as we move into secondary structure. Secondary structure is when a protein begins to take on its characteristic three-dimensional properties. We need to transition from primary structure into secondary structure by what is called folding. Now proteins can fold, the amino acid sequences can move around, they can bend, but they're also going to need to form bonds in order to stabilize the secondary structure. So let's take a look at the chemistry, the stereochemistry of a primary structure to understand how this actually works. So I'm just going to draw a couple of amino acids here linked together. Here's our amino acid 1, amino acid 2 side chains. We have a hydrogen here. We have a hydrogen here. We have one here on the nitrogen and one on the nitrogen. Now recall, when we link two amino acids together, we're forming a peptide bond. The peptide bond has a very unique property in that it is a partial double bond between the oxygen and carbon and nitrogen. So this is more accurately written as a partial double bond. Now recall, rotation around a double bond cannot occur. Because this is a partial double bond, this particular structure will remain rigid. In other words, rotation around this bond cannot happen. Now that means every peptide bond that is formed in the linear structure of a protein in the primary sequence is going to have a planar effect. What's also important is that these particular bonds around the nitrogen and alpha carbon and the carbon and carbonyl, these are free to rotate because these are single bonds. These angles can be measured, angle of rotation. This is called phi. This is called the psi angle. And the angles at which these can turn will largely be dictated by steric properties of the R chain. So if you have two bulky groups here on these R chain, they are going to want to be as far apart as they can. And so moving these away from each other means rotation of these bonds, primarily the phi and psi, into a configuration and angle at which these R chains are moved away from each other to prevent any unfavorable interactions. Now, to move from primary to secondary structure, we need to consider the planar properties, the immobile properties of the peptide bond, in consideration with the phi and psi angular changes, due to the R chain groups. Once we consider these, we can now draw potential secondary structure. Because recall, these amino acids are all L, levorotary configuration. That is going to give the secondary structure a right-hand turn, looking from N-terminus to C-terminus. Each turn is approximately 4 amino acids long until we come back to the next point. Now, as you can imagine, this can be a very flexible structure. So we want to stabilize the secondary structure. So what stabilizes this? There are 2 chemical properties that stabilize. One is that, recall, the nitrogens in each one of these turns has a hydrogen on it. As you turn here, the 4th amino acid around the turn will be a carbonyl. Now recall, because of the electronegativity of the nitrogen and oxygen, these are going to be electronically shifted so that we have a partial positive charge on the hydrogen and a partial negative charge on the oxygen. These will form hydrogen bonding. This will happen progressively down the chain. Now, each one of these bonds is weak. This is not a covalent interaction. However, the accumulation of all of these down this entire looped structure will be additive and give the stabilizing. The other thing to keep in mind is that when a primary structure folds up into this type of secondary structure, the objective is to move the R chains out of the way as quickly as possible so that there is no steric hindrance or steric interference between them. Many of these R chains are polar or charged. That means that we can form hydrogen bonds with water, and if the side chain is charged, we can form salt bonds. Sodium, potassium, that is also in the aqueous environment. That will contribute bond energy to these R chains to help stabilize the structure. Now, this particular structure I've drawn here is called an alpha helix. Now, an alpha helix is not the final structure of a protein. Remember that many of the proteins we're talking about have enzymatic activities, which means there needs to be a 3-dimensional binding cleft in an enzyme. 3 dimensions means that now we have this simple structure needs to come together to form a more complex structure of an enzyme. This is called tertiary structure. Additional folding must occur. The final product of tertiary structure will be our monomeric protein unit, and it's this structure that will give us our final structure of our protein. For example, if this is the final goal, of our folded protein, this would be called tertiary structure. And notice that we have a cleft. So if this is an enzyme, substrates now have a place to form bonds with the enzyme and then be subsequently converted and catalyzed into product. So how does tertiary structure come about from secondary? Tertiary structure is primarily interested in these R chains. So if we have an alpha helix with R chains coming out the sides of the helices, for example, we could have an alpha helix form in part of a protein, and then it could potentially loop back on itself and form another alpha helix. Now picture these R chains forming bonds. If they are hydrophobic, two opposing hydrophobic bonds will interact by hydrophobic forces. Also van der Waals, contributing to this bonding. What if these R groups are charged, such as a lysine opposing a glutamate? These can form ionic bonds. Likewise, if we have opposing polar groups with partial charges, we can have polar groups interact. Now again, any one of these types of bonds are non-covalent, so that they're not very strong, but when you have many of them that line up, then you have a very strong interaction. If this is a sufficiently strong interaction, this tertiary structure, which is a combination of secondary structures, will be stable. Now what happens if these interactions here are insufficient to hold these two alpha helices together? Well, then they will come apart. The alpha helices will maintain, but the total tertiary structure will collapse. Other alpha helices that might be present in the folded protein might have better interactions. Better means more bonding energy to lend greater stability. So folding is essentially what's called a sampling. Alpha helices will come together, sample each other to see if they have complementary R groups to form the proper bonds and interactions. If this is insufficient, they will come apart and test another alpha helix. Now there are a couple of other considerations here. This loop structure here. If we had one long alpha helix generated by the primary structure of the protein, this could potentially form a very rigid structure that could not fold back on itself to do this sampling to form tertiary structure. So periodically we need to break the alpha helix. Now we're not talking about breaking chemical bonds such that we are breaking covalent bonds. What we are doing is we are interrupting this sequence of turning of the primary structure into secondary. And the primary way to do that is to introduce the amino acid proline. Proline is an amino acid, which means that simply it is connected between the alpha carbon and the nitrogen is connected by a ring structure. So what this simply means is that the rotation that normally would occur between the nitrogen and alpha carbon, which is called your phi angle, cannot occur. Therefore, we disrupt the normal periodicity of turning of the amino acids to form the alpha helix. So proline is considered a helix breaker. Once the helix is broken, we get into what's called a random coil. The random coil has no secondary structure to it, but if we have the proper amino acids beginning to fold again into secondary structure, we can begin to form another alpha helix downstream from the breaker. By having a random coil introduced between the two alpha helices, we can now have sufficient flexibility in order for these two alpha helices to sample each other to see if their interactions are appropriate for a stable tertiary structure. Now, the other secondary structure that we need to address is called a beta sheet. And a beta sheet is a very different secondary structure. This is a linear structure that does not fold up into a tertiary structure. Notice the way I've drawn this sequence of peptide here by putting the R chains on opposite sides. Here's our peptide bonds. These are 3 amino acids. The way a beta sheet turns is it actually lies flat. It does not turn into an alpha helix. It lies flat so that the R chains are essentially in opposite directions. So the peptide backbone, which is the long linear structure of nitrogens and carbons here, is flat. If we were to turn this on its side, this would be the nitrogen-carbon backbones, and the R chains would be sticking up and down based on the rotation. What this allows to do is another beta sheet come in and potentially have R chain interactions. If, again, the side chains of these amino acids are complementary. So if they are hydrophobic, that is complementary. If they are charged, opposite charges. If they are polar, forming hydrogen bonds. These are each called beta sheets. And these can form very, very stable structures and very strong structures. A good example of a protein that is largely, almost exclusively beta sheet is silk. If you've ever tried to take silk thread and stretch it, it's virtually impossible. Compare this silk protein with albumin, which is a blood protein of ours. It's the highest concentration of any protein in the blood, albumin. this albumin is an alpha helix. So the secondary structure that can form from the primary structure is largely dictated by the sequence of amino acids and how these proteins line up with their R chains. Now the final topic I'd like to discuss is protein folding. What is the thermodynamic considerations of protein folding? And the best thought that I've seen on this in textbooks is a funnel effect. If we were to measure energy of substances in this funnel, energy would be highest at the top of the funnel. This would also be considered the greatest entropy entering the top of the funnel. If we look at another measurement called stability, stable conformations of proteins, this will decrease because we are forming the structure. So this will become more stable at the bottom. Now, how does this consideration determine protein structure? Picture a primary sequence of a protein. The primary structure of a protein will enter this funnel, and it will be at its highest energy state. Because it has not yet formed all of those bonds that will lend to greater stability. It also has its highest level of entropy because of the randomness of this long linear chain. Now, as we move into this funnel, we are going to generate secondary structure. Secondary structure is going to be at a reduced energy state because of the bond energy we are forming within these alpha helices between the hydrogen bonds, between the R chain interactions with the solvent and the environment. This is also, by having more of these bonds, increasing stability and decreasing entropy. Now these alpha helices, for example, will sample each other, looking for R chain interactions that will increase bond strength in the structure, and eventually we will reach tertiary structure, which is the final folding structure of the protein. This will be the most stable form. So where does protein folding occur? It's in this region here. It's in the region where secondary structure is continually sampling to find the greatest level of bond formation, those non-covalent bonds, to lead towards stability. Now, protein folding, as you can imagine, is a very complex process. There are many proteins that can fold by themselves. One example is RNase. RNase is a classic protein that can fold spontaneously. It needs no assistance. It can snap quickly into its final form. Some proteins need assistance. And there are small proteins that are located both in the cytoplasm for cytoplasmic protein assistance, as well as in the lumen of the endoplasmic reticulum for secreted and membrane protein folding assistance. And these are called chaperones. And what these chaperones are is you could picture them as a very large Pac-Man. They are there to assist in protein folding. So you can picture maybe an alpha helix that binds onto one area here, a random coil, and another alpha helix of the protein. The job of the chaperone is to assist in these two domains coming together to increase the speed of folding. If these were to find themselves through mass action, it could potentially take hours, days, weeks to find the lowest energy form, the most stable form of the tertiary conformation. But the chaperone acts as a catalyst to bind secondary structures and bring the appropriate secondary structures together, which increases the speed of folding. Now the final information that we will examine even further later is tertiary to quaternary folding. This best example is hemoglobin. We'll be examining the properties of hemoglobin in another video, but suffice it to say that tertiary structure is a monomeric single protein, single polypeptide, and when these single polypeptides come together and interact by having affinity on their binding surfaces, for hemoglobin, 4 subunits come together to form hemoglobin, and this final structure is called quaternary structure.